like you just heard, I'm going to give you an update on ff19SB, talk a little about how we designed it, and then some more recent testing that we've done. Okay, so ff19SB was developed mostly because of problems that we ended up seeing in ff14SB. Lots of people have used ff14SB, so there are many good things. Here's one example from a couple of years ago in my group where we were looking at protein-protein binding affinities and how they change with amino acid mutations, and found that we could reproduce pretty accurately the changes between different amino acids and their impact on the binding affinities compared to experiment. And so that was a good sign that at least our energetics were reasonable. This is a more recent one that's ligand binding to proteins using F14SB into 3P and the GAP small molecule force field. This one's not out, but there's a preprint out on ChemArchive if you're interested. And these were all on ligands where we had to do ring opening and closing or linker contraction or things that would normally be considered very challenging. And by using some restraints, we showed that even though these were pretty large transformations for an alchemical calculation, that we again got pretty good correlation with experiments. And maybe a little more relevant to the things that I'm going to show you today, we had shown with our GV implicit solvent model that with F14SB, we could get proteins of a variety of secondary structures and sizes to fold to structures that were pretty accurate. And so this shows that the secondary structure balance is really good. There's all beta proteins, all alpha proteins, mixed ones. And so that was reasonable. It showed us at least, not that this is how you would do protein folding or structure prediction, but that our energy functions were reasonable on these time scales. But when we started to try to take some of those proteins and say, let's make some amino acid substitutions and see if they impact the stability the way the experiment says, there we found very poor correlation. We would make some changes that were supposed to make things more stable and they would get less stable, and the other way around. And so eventually we ended up tracking that down to some things that originally we saw from Robert Best, calculating helical propensities for the different amino acids and comparing them in the experiment. So I'm going to show you a bunch of these plots, and the helical propensities for all the different amino acids are dots, and then our estimates from NMR are on the x-axis and our MD simulations on the y-axis. So every one of these dots from the simulation point of view comes from 12 simulations of about 3 microseconds in explicit water. So overall, every one of these plots is about a millisecond. This kind of testing is really expensive. And so we saw that there was a lot of scatter, and we thought, well, maybe that just means that, we're not doing a perfect job with the experiment. But in the cases that we checked, the errors in some of these helical propensities mapped back to the errors that we were getting in our stability changes pretty well. We would make mutations in a helix, for example, to try to stabilize something like in trip cage, and instead of stabilizing the protein, they would destabilize it. And that's because of this anti-correlation in some of these. So we thought, well, maybe this is really just our force field not doing a great job. So we switched out to another variant of our older ff99SB force field, and found that that didn't really help things very much. So we thought maybe it's the water model. We switched to the Nuclear Labs OPC that you heard a little about, their polarizable one earlier. And this didn't work well at all, other than these outliers for the negative charge ones. These really almost all have the same helical propensity. And so we had no sequence dependence at all. So we looked at a wider variety of combinations, different water models, different ways of doing the force field, ff15 IPQ with a different charge model, force balance force field and water model, and really didn't see any improvement in these. That there was just a lot of scatter and not much of agreement with the experiment. Some of these have outliers that in some cases the outliers are the positive charged amino acids, but very often alanine is an outlier either being much too low or much too high compared to the experiment. So we decided to go back and try to do a better job on this. And since we were going to redo our fitting for the protein backbone, we thought that we would try to take care of a lot of different limiting factors together. One, all of our previous fitting had been to gas phase quantum, and we thought that wasn't a great target because we go to a lot of work in this non-polarizable model to make sure that our charges look like solution phase charges, but then our dihedrals are fit to gas phase calculations, and we end up counteracting some of our polarization with the dihedrals. And so that's not really what we want to do. The ff19SB backbone, which is what we used in ff14SB, had only been fit to minima, and there were gas phase minima, so we decided we needed to fit the whole surface. Phi-sci coupling, this really came from Alex and work in the charm community on CMAPs. And then, like I talked about from some of our correlations, we decided that alanine wasn't a very good model. It's an outlier in a lot of properties, just like glycine and proline are. And so we decided that leucine is really more representative with a gamma carbon. Most of our amino acids have specific maps, but when we don't have one, we use leucine. And then the last thing, based on work from Alexi and others, we decided not to use TIP3P for validation like we did in ff14SB because it has a lot of issues of its own. So we're using a 2D CMAP like is done in CHARM. And this on the left is a Ramachandran map, phi and psi, for alanine dipeptide in the gas phase. And on the right, in our particular choice of quantum that we use for ff19SB. And the gas phase, not surprisingly, is really quite different from the solution phase map. And so when we fit to the gas phase, as we did in ff99SB, we needed to use the tetrapeptide in order to even make the helical conformation a local minimum. But now we're going to fit the whole map, and we're going to do it to this solution one that I show you on the side. We did a little bit of work trying to figure out the best balance of performance and accuracy for our quantum. We needed about 10,000 optimizations, so we needed it to be relatively fast. And this is what we settled on with SMD solvent. And the error compared to the higher level quantum isn't that bad. And I think that that's probably much bigger than other errors we're making in our other force field. This has kind of been an Amber philosophy for a long time, is not to try to spend a lot of time working on things that aren't your limiting errors. This really came from Peter Coleman. And so why would we need coupled phi-sci corrections? I know, that this has been around for a while in the charm community. Like I said, Alex is the one that talked us into doing this. And this is just from us trying to figure out, is this a two-dimensional correction so we have more parameters, or is this something that we actually do need to be doing? And so on the left and right, what we did was on the Ramachandran map, we took all of the structures that we had scanned, and then for every one of them, we used RESP to calculate what our charges would be that we could get from this. We could also do dipole calculations or things, but it was just quick to say, well, what if we had conformation-dependent charges that we had fit? And you can see all of these diagonal features. Here's for the carbonyl, here's the NH, these are both the backbone amides. And as you vary phi and psi, there's coupling between these. And so the polarization of the backbone should be backbone dependent. And as we heard in the last session from all of our polarizable charge models, that's a better way to do things. But in the absence of that, this is one of the reasons these interactions between these amides, as you vary the backbone conformation, this is one of the reasons that we think we need these coupled corrections, that at least in a fixed charged model kind of way, we can include some of these interactions into our dehedral parameters. And so that's why we're using the CMAPs. Okay, I mentioned that we're using solvated quantum. We did some tests to see if we really needed to do that, or if that was just, an idea that that seemed good but didn't make a difference. On the left here, the top is our QM plus SMD. So this is our target 2D free energy surface for alanine dipeptide. And then the bottom is the ff19SB with GBSA. I'll get to that in a minute. And because we're fitting this exactly and there's only 2 degrees of freedom, that's the map we get, and we can reproduce it very well. But if we did this in the gas phase with our quantum and did that same scan and fit our model in the gas phase to this, so we had a CMAP that was based on the gas phase, and then tried to use that with the same GBSA model, you can see that the map we get out has some features that really don't look very good at all. We have an alpha helical basin that's much too stretched into -phi values. We have too much stabilization over in the positive phi, and this did not work very well at all. And so we decided that we really did need to be fitting against having the solvent and the QM level. And so what we do is we've got the quantum with SMD, and we use ff19SB. When we do the fitting, we use GBSA so that they're both an implicit solvent, similar implicit solvent models. And so we decided to see whether our force field was going to be really dependent on the solvent model. We don't want to have a force field, and in the end, we've trained it with GB, and so it only works well in GB. This is alanine dipeptide down at the bottom, and for small molecules, they're really not very sensitive to the kind of solvent model that you use. And so these are Ramachandran energy maps for the alanine dipeptide And what we're doing here is we're varying just the solvent model that's included with the MM. So we tried PB with 2 different radii sets, we tried 2 different explicit water models, and we tried 3 different variations on GB. And it's really difficult to tell these apart. They have very small effects. And this plot over on the right is just the matrix of deviations, showing you that typically they're on the order of an average of a few tenths of a kcal different from each other. The explicit solvent models tend to be pretty similar. The GB models are pretty similar, but overall, there's not a lot of difference in the accuracy. So we decided to go with GBNEC2, which is our one that of the GB models that we have, we think is the most recent and the most accurate one. Okay, so we're including solvent in the MM, we're including solvent in the QM, we're doing the 2D mapping, and the next thing was that we decided that we were going to do amino acid dependent corrections instead of just basing them all on something like alanine. And so here, the middle column is from the PDB coil library. So this is non-secondary structure data extracted from things like loops and high resolution structures. And this is ff19SB on the right. These are populations from MD simulations, and these are kind of like populations from the PDB if you want to think that those are like that, but they're clearly not Boltzmann weighted. So we only really look at the contours and not the depths of these minima. And so top is alanine, bottom is valine. The PDB tells us that when you go to beta branch valine, you get a smoothing of the PP2 to beta transition. You get a narrowing here of the alpha minimum. And when we look at ff19SB, we don't see those features at all. They're really almost indistinguishable. And I think that's because our parameters are just too generic. We didn't have phi-sci coupling, and they all used calculations based on alanine. So it might be that these are features that only show up in proteins, not in dipeptides. But when we looked at our quantum data, we found that they were consistent with the PDB, that we've got a bigger separation of the beta and the PP2 in alanine than we do in valine, where it's flatter and sort of U-shaped. And then instead of having this diagonal basin for the alpha helical, we've got something that's more rounded and narrow, just like you see in the PDB. So we thought that these probably do need different corrections for them because we're not getting that coupling between the backbone and the side chain right, or it's possible that the different side chains are changing the impact of polarization on those amides because now we've got a side chain in between those 2 amides. But we decided to go with the amino acid specific CMAPs. So we have 16 different ones, quite a few of them. The other ones besides those 16 use leucine, and leucine is what we show or what we use when we have a modified amino acid. And so now this is ff19SB on the right. I showed you these before, and you can see that for glycine and alanine, we get basically a perfect match to the QM because that's what we're fitting to. And we get these features that we didn't see in ff19SB. We've got some phi-sci coupling in our helical basin, and there's just a better shape overall in ff19SB. But those are fit to the quantum, so they should look exactly like this. And when I first started talking about this with Alex, one of the things that he suggested that we really needed to do was to look and see for things like not alanine, how transferable the CMAPs are. Are they going to be different for different rotamers and so on? So can we use a CMAP calculated for one rotamer and then use that CMAP for another rotamer? And so here for valine, we've got the rotamer that we trained against, ff19SB, again, QM and ff19SB match exactly because that's the training data. But then when we change our rotamer, you can see in ff19SB, there's not a lot of difference, except maybe the alpha helical basin gets a little bit deeper, and these 2 minima over in positive phi merge into 1 in the middle. But that's not what the quantum says. The quantum says that we should actually change the shape of the helical minimum because this beta branched amino acid affects our helix, and that this lower basin really should disappear and keep the upper one. And so this is our training data in the top, but you can see when we change the rotamer that we reproduce those differences in the backbone potential pretty well, showing that CMAP is going to be transferable from one rotamer to another. And we did that on quite a few amino acids. So I don't think that these CMAPs are very sensitive to the rotamers. This is more quantification of that data. Lots of these are training, but let's look over here at the glutamate. What we're doing is we're going from the minimum on the quantum surface up in energy, and we're getting to that energy cutoff and saying, okay, for all of the structures below that energy, let's calculate the error versus the QM, the average error. And these orange curves are for ff19SB and the blue are for ff14SB. And so here we've done training on this rotamer, so we have a really good match to the whole surface. ff19SB doesn't, but when we change the glutamate rotamer, we can go up to about 10 kcals per mole in energy and still have about a half a kcal error in all of our conformations relative to the quantum. And so for all of our amino acids, we get this behavior where even for things that are outside our training set, we're reproducing pretty well what those are. These are all trained in a dipeptide. In ff19SB, we had used a tetrapeptide, like I mentioned earlier. And so here's our CMAP on the dipeptide. They match exactly. And now this helical basin here doesn't actually have a hydrogen bond. It's just a helical local conformation of the backbone, but no real alpha helix. So we went to the dipeptide, kept everything, thank you, kept everything helical except for one amino acid and redid the scan, the 2D scan in the quantum. And now you can see that we're adding onto a helix. The helical basin becomes much deeper, but when we take the same CMAP from the dipeptide and use it in that system, we find that it's very transferable. And so there's no real errors in the strength of that hydrogen bond compared to the other errors that we have in our system. So we can move from the dipeptide, I think, pretty confidently to a larger protein. These are just rotamers for the side chain conformations, showing you that we do the side chain chi-1 rotation for one backbone conformation, and the match is good. And then we change the backbone and do that again. And we also match the quantum pretty well for being a relatively simple model. Okay, so how do we do now compared to the experiments? Here, I showed you this earlier where we didn't reproduce the coil library changes. Now we do a much better job of reproducing the change in valine and other amino acids. It gets a flatter transition, a narrower alpha, and so on. And then we wanted to look at different water models. You probably saw this earlier in the OPC talk, where they've quantified errors in different water models. We calculated the helical propensities in ff19SB in all of these different water models. And you can see that some of them, like TIP3P, are actually quite poor. But by the time we get to OPC water with ff19SB, the agreement with the experiment is really quite good. And so generally we find that as our water model gets better, the correlation between the experiment and the protein force field gets better. And so I think that we're at the point for this test, at least, where we're limited by the water model and not the protein backbone. But this is a very simple test of helical propensities. What's the difference between the water model and the protein force field? When we compare ff19SB and OPC and in TIP3P, you can see the correlation's not too bad, but the TIP3P gives us things that are much too helical. And that's probably because TIP3P favors compact structures with a weaker dispersion interaction. And so that results in secondary structure biases as well. Here now we've kept the same water model, but we're switching the force field, and you can see that the slope is near 1, but there's a lot of scatter. So the force field gives you better correlation for the amino acids, and the water model affects the overall trends.
2 minutes.
Perfect. We've just finished new amino acid libraries for both ff14SB and ff19SB. I know ff19SB with its amino acid specific CMAPs isn't in a lot of other programs. ff14SB is. So we released libraries for both of these that are part of Amber tools, lots of phosphorylated amino acids and other ones that are modified for experiments. And we had other deprotonated amino acids, and now we've got a tyrosine there as well. So I think I'll skip these. You've seen really everything that I went over. And this was a collaboration with Chin Wu at Brookhaven Lab, who was the quantum side of things. And I want to thank everybody in my lab, especially Xuanxiang, who is now at Eli Lilly. He was the grad student that led this. And thanks for your attention.